Welcome to the deep dive. Today, we're getting into a really fascinating stack of sources you sent us.
Yeah, it's quite the mix, ranging from like 19th century medical history to some pretty intense modern gastroenterology debates.
Right, and our mission today is to explore the ultimate boundary between the outside world and the inside of your body.
Which is such a wild concept to think about.
It really is because, when you think about that boundary, you probably picture your skin.
Sure, yeah.
It makes intuitive sense. It's the physical wall of the castle, right?
Yeah.
But biologically speaking, if you swallow a penny, that penny is still technically outside your body.
Exactly. It's simply traveling through a hollow tube that just happens to be folded up inside your abdomen.
Which is so weird to conceptualize.
It is. The true boundary, the actual place where the outside world crosses over a cellular barrier and officially becomes you, is deep inside.
And that is exactly what we're dissecting today, the small intestine.
Yes, the ultimate gatekeeper.
But before we get into how it actually works, we have to clear up some major misconceptions, starting with, well, the name itself.
Oh, absolutely. The name is terrible.
Right. So the small intestine runs from the pyloric sphincter, which is just the exit door of your stomach, down to the ileocecal valve, where it meets the large intestine. But calling it small is highly misleading.
Yeah, small refers strictly to its diameter. It's only about 2.5 to 3 centimeters across.
Okay.
Whereas the large intestine is much wider, about 6 centimeters. But in terms of length, it is the undisputed giant of your gastrointestinal tract.
It's massive.
Right. In a living, breathing adult, constant smooth muscle tone keeps it coiled up to about, say, 3 to 5 meters long.
Which is already a ridiculous amount of tissue to fit inside a human torso.
It is, but what's really illuminating is what happens at autopsy. When that smooth muscle tone is completely lost, the tissue relaxes and it just stretches out to 6 or 7 meters.
Wow.
Yeah, so it's several times longer than the large intestine.
Okay, let's unpack this. Because we have this massive coiled 20-foot tube, but the physical length isn't even the biggest paradigm shift in our sources.
No, not at all. The way science thinks about what happens inside this tube has been completely rewritten over the years.
Right. If you look at 19th century physiology, scientists viewed the gut basically as a passive filter.
Yeah, they thought of it like a coffee filter or like a sieve.
Exactly. The assumption was that you put food in and nutrients just sort of seep through the intestinal membrane.
Just moving naturally down a concentration gradient. They really believed it was this completely passive process.
Like molecules just following the path of least resistance into the blood.
Exactly.
So it's less like a coffee filter and more like, what, an active conveyor belt?
Oh, it's much more aggressive than a conveyor belt.
Yeah.
That whole 19th century idea was totally overturned by the biochemical revolution in the mid-20th century.
Right.
We now know that the small intestine is an actively driven, incredibly energy-consuming engine. Yes. It uses the exact same kind of ion pumps that power your nerve and muscle cells to forcibly drag nutrients into your body.
Even when it has to fight against concentration gradients.
Especially then. Moving from this idea of a passive sieve to a highly selective active engine is, it's one of the most profound yet quiet revolutions in our understanding of human anatomy.
An energy-burning engine that spans 20 feet. That is wild.
It really changes how you look at digestion.
But to understand how this engine works, we really have to look at its architecture. Because there is a serious engineering problem here.
Oh, massive spatial constraints.
Right. You have to fit an absolutely massive absorptive surface area into this compact tube so you have enough room to grab all those nutrients before they, pass on through.
And nature's solution to that spatial problem is just a masterpiece. It's this fractal-like nesting, basically a 3-tiered folding strategy.
Let's walk through those tiers. So
Yeah.
fold number 1, the plique circularis.
Also known as the valves of Kirkring.
Right. These are permanent circular folds in the wall of the intestine itself. If you look at them, they look almost like speed bumps lining the inside of the tube.
And mechanically, they serve a very similar purpose to speed bumps. Those folds physically slow down the chyme.
The chyme being that highly acidic, partially digested food arriving from the stomach.
Exactly. By slowing that fluid down, it churns the mixture and gives the intestinal lining way more time to extract the nutrients.
Makes sense.
And more importantly, those visible folds immediately multiply the surface area of the tube by 3.
But that's just the macro level, right? On top of those folds, you have fold number 2, the villi.
Right, which early microscopists actually discovered back in the 17th century.
Which is amazing. They're these tiny finger-like projections, maybe a millimeter tall, just carpeting the entire inside of the tube.
Like velvet.
Yeah, exactly. And they add another tenfold increase to the surface area.
And then, if you zoom in even further, down to the level of a single absorptive cell, an enterocyte, you find fold number 3.
A microscope law.
Yeah. On the tip of each cell facing the inside of the gut, there are thousands of microscopic projections called microvilli.
And under a light microscope, this looks like a dense, fuzzy border, right?
Exactly, which is why anatomists call it the brush border. And that cellular folding adds yet another 20-fold increase to the surface area.
Okay, let me do the math on that real quick. 3 times 10 times 20. And this actually brings up a massive math error in our sources that completely changed how I picture this organ.
Oh, the textbook error. This is a great one.
It's crazy. If you open almost any biology textbook from the last 50 years, it will tell you that this total surface area is about 250 to 300 square meters.
Here's like this tennis court statistic.
The tennis court. Every medical student, every biology undergrad gets taught this. Your small intestine has a surface area of a tennis court.
Yep. Repeated everywhere.
But wait, if scientists have had microscopes for centuries, how did a physical measurement like that go unchecked for so long?
Well, it's a fantastic lesson in how, unquestioned assumptions just get baked into scientific dogma. Right. The original estimates, which were made decades ago, treated those complex, squishy, irregular biological folds as if they were perfectly rigid, flat geometric planes.
Like they were measuring a parking lot.
Essentially, yeah. They calculated the theoretical maximum if you ironed everything out perfectly flat. It wasn't until 2014 that Swedish researchers Herbert Hirlander and Bjorn Fendrix finally decided to check the math using rigorous stereological methods.
Stereological meaning they actually accounted for the fact that these are 3D compressible irregular structures interacting dynamically in a living body.
Exactly. They used advanced microscopic imaging to measure the actual functional tissue, and they found the true absorptive surface area is about 30 square meters.
30 square meters. That is roughly half a badminton court.
Half a badminton court, not a tennis court. The textbooks were off by nearly an entire order of magnitude.
That is just wild.
And yet, because the size of a tennis court was such a sticky, memorable fact, it was taught to generations of doctors without anyone going back to the first principles of anatomy to verify it.
Still, I mean, half a badminton court is an astonishingly massive factory floor to fold up inside your abdomen.
Oh, without a doubt. It's incredibly dense.
So now that we know the true size of this factory floor, how did the machines actually pull the nutrients across the border? Because earlier we established this is an active engine, not a passive sieve.
Right. To understand the mechanism, we really have to look at how stumped early doctors were by this. For over 1,000 years, going back to the physician Galen in the 2nd century, medicine relied on this vague concept of concoction.
Concoction.
Yeah, they literally thought food was just magically transformed by body heat.
Wow.
Even when the great anatomist Vesalius corrected a lot of structural mapping errors in 1543, he couldn't explain how absorption happened. He had a scalpel, but he didn't have cell theory or biochemistry.
He could map the highways, but he had no idea what was under the hood of the cars.
Beautifully said. The real breakthrough didn't happen until 1960. A physiologist named Robert K. Crane presented a paper proposing a mechanism called co-transport. He hypothesized that a specific membrane protein on the brush border ferries a sodium ion and a glucose molecule together across the intestinal wall.
Let me see if I understand the physics of this. Your cells have these pumps, the sodium-potassium pumps, so they constantly burn energy to push sodium out, keeping sodium levels very low inside the cell.
Sure.
That creates a massive vacuum or gradient. Sodium desperately wants to rush back into the cell, like water building up behind a dam.
Exactly.
So Crane realized that the intestine uses the sheer force of that rushing sodium to physically drag glucose in with it, even if the body already has plenty of sugar.
That is the absolute genius of it.
It's kind of like sneaking into an exclusive concert by tightly gripping the jacket of someone who already has a VIP pass.
That's a great analogy. The sodium has all the momentum, and the glucose just catches a ride through this door called the SGLT1 transporter.
And then the glucose exits out the back of the cell into the blood via another door.
Yes. And this isn't just an academic quirk of cellular biology either. Crane's co-transport model is considered one of the most consequential ideas in 20th century medicine.
So what does this all mean?
Well, it directly explains how we can save people from one of history's deadliest killers, severe dehydration from diarrheal diseases like cholera.
Wait, how does molecular transport connect to cholera?
So cholera toxins severely damage the gut lining, right? Causing it to leak massive amounts of water. Historically, patients died because their intestines were too damaged to absorb plain water. They would just expel it.
It's just terrifying.
But because the sodium-glucose co-transporter forces the body to absorb sodium and glucose together, scientists realized they could hack the system.
How do they hack it?
If you give a patient oral rehydration therapy, which is a solution of precise amounts of salt and sugar in water that specific SGLT1 pump activates, it violently drags the sugar and salt into the tissue.
And water naturally follows via osmosis.
Exactly. It completely bypasses the damaged tissue.
So by just adding a specific ratio of sugar to salt water, you force this molecular engine to turn on, pulling water back into the body.
Yeah, that simple mechanism has saved tens of millions of lives since the 1970s.
That is incredible.
It works because it relies on the fundamental, hardwired mechanics of the small intestine. But it's important to note for you, the listener, that not all sugars get this VIP co-transport treatment.
Right, the sources mentioned fructose is a glaring exception to this.
Yes, fructose uses a completely different door, a transporter called GLUT5, and it doesn't use that sodium-powered buddy system at all.
It's totally independent, which means it moves much, much slower.
And that biological bottleneck has very real, very uncomfortable everyday consequences.
Like what?
Well, if you overwhelm that slower GLUT5 transporter by, say, slamming a giant high-fructose soda or drinking a massive amount of apple juice on an empty stomach.
All that unabsorbed fructose just sits in the intestine.
Exactly. It acts as an osmotic load, drawing water into the gut where it ferments and causes severe bloating and diarrhea.
Your body literally can't process it fast enough, so the factory line backs up.
Precisely.
And it's fascinating that proteins have their own specific doors too, right?
Mm-hmm.
Like the peptide-1 transporter for amino acids, which is also heavily sodium dependent.
Yes, very specific entry points.
So sugars and proteins go through their specific doors on the brush border, get into the cells, and get dumped straight into the blood capillaries. That makes sense, since blood is mostly water. But what about fats? Because oil and water don't mix.
Yeah, fats require a completely different, highly specialized pathway. It's essentially a microscopic assembly line. First, the dietary fats you eat are emulsified by bile.
Which comes from the liver and gallbladder.
Right, the bile acts just like dish soap breaking up grease in a frying pan. Then, an enzyme called pancreatic lipase breaks those fats down into smaller free fatty acids so they can diffuse across the cell membrane.
But here's the part that really blew my mind in the sources. Once those fat pieces are inside the intestinal cell, the cell rebuilds them.
It does.
It stitches those pieces back together into triglycerides, packages them with cholesterol and a protein wrapper, and creates these massive cargo ships called chylomicrons. But why go to all the trouble of breaking a fat down just to rebuild it?
Well, because a massive glob of fat can't cross the cellular barrier. It must be broken down to enter. But once inside, it can't just be dumped into watery blood, or it would clump up and cause a fatal blockage.
Oh, that makes sense.
By packaging it into a chylomicron, the cell creates a water-soluble transport vessel. However, these chylomicrons are simply too massive to fit into the tiny blood capillaries inside the villi.
So they need a completely different highway.
Precisely. Inside every single villus is a specialized lymphatic capillary called a lacteal.
A lacteal.
Yeah. The massive chylomicrons enter these lacteals, travel up through your lymphatic system, and bypass the liver entirely. They eventually get dumped directly into your general bloodstream near your heart via the thoracic duct.
Think about that. Sugars and proteins go straight to the liver to be strictly inspected and metabolized. But fats take this secret lymphatic highway and get dumped straight into general circulation.
It's a very different logistical route.
That kind of highly specialized routing makes me wonder, does this entire 20-foot tube do the same job everywhere? Or are there different departments?
Oh, it is heavily compartmentalized. The small intestine is divided into 3 distinct functional regions. First, you have the duodenum. It's incredibly short, only about 25 centimeters long.
But think about what's arriving from the stomach, though. It's a highly acidic, dangerous slurry. If that just dumped straight into the delicate lower intestine, it would burn a hole right through the tissue.
Exactly why the duodenum is structured the way it is. It acts as a chemical neutralization chamber. It receives that acidic chyme and immediately neutralizes it with a massive flood of bicarbonate.
Like putting out a fire.
Yes. It's also where the bile and pancreatic enzymes are dumped in to initiate the heavy chemical breakdown.
So it's the prep kitchen. And once the food is neutralized and prepped, it moves into the jejunum, which is roughly the next 2.5 meters.
Right?
This seems to be the heavy lifter of the whole operation. It has the tallest villi and handles the vast bulk of nutrient absorption. Whether it's sugar, amino acids, or those fats we talked about, the jejunum is grabbing them.
Which leaves the final and longest segment, the ileum. And the ileum is the absolute specialist of the gut.
Wait, so if it's divided into these strict departments, what happens if one goes offline? Like, if a surgeon has to remove part of the jejunum due to trauma, does the body just stop absorbing food entirely?
You'd think so, but here's where the engineering gets really resilient. The intestine has a built-in functional reserve. It can adapt.
Really?
Yeah, if you lose a significant portion of the jejunum, the ileum can actually change its cellular structure to pick up the slack for basic nutrients.
Wow.
But the reverse is not true. The ileum is completely non-redundant. It performs two critical jobs that the rest of the small intestine simply lacks the hardware to do.
I'm looking at the notes here. The first is the active reabsorption of bile salts.
Yes. Your body recycles about 95% of its bile salts in this incredibly efficient closed loop called enterohepatic circulation. That reabsorption only happens at the very end of the ileum.
And the second unique job.
The absorption of vitamin B12, which is crucial for nerve function and making red blood cells. B12 can only be absorbed in the ileum, and only if it has already been bound to a special protein from the stomach called intrinsic factor.
Oh, and the ileum also houses Peyer's patches, right?
Yes, dense clusters of immune tissue.
It's a great reminder to you, the listener, that this isn't just a food tube. It's one of the largest immune organs in your body, constantly sampling what's passing through to watch for pathogens.
Absolutely.
Because every single part of this tube has such a highly specialized job, bypassing it has profound systemic consequences.
And the medical field learned that the hard way. In the 1960s and 70s, a surgical procedure called the jejunal bypass became popular for treating severe obesity.
The logic seems brutally straightforward, right? If the food doesn't travel across the factory floor, you don't absorb the calories, and you lose the weight.
And the patients did lose weight dramatically. But the procedure was a physiological disaster. The surgeons drastically underestimated the specialized, non-redundant work of the ileum.
Because they skipped over it.
Right. Because patients weren't recycling bile salts, they developed severe fat malabsorption and horrific kidney stones.
Oh, wow.
Because they couldn't absorb B12, they faced neurological damage. Even worse, the blinded bypass sections of the intestine became breeding grounds for bacterial overgrowth, leading to liver failure.
That is awful.
It was. The procedure was eventually abandoned for more careful surgeries like the modern Roux-en-Y, which deliberately preserve essential intestinal function.
It's a classic case of thinking we can outsmart a 30-square-meter engine without really understanding all the underlying gears. And speaking of understanding the gears, there's a fascinating natural experiment from World War II that unlocked another massive mystery of this organ, the Dutch hunger winter.
Oh, this is such an important piece of medical history. In the winter of 1944 to 1945, the German occupation blocked all food supplies to the Western Netherlands.
It was a brutal, devastating famine.
But amidst the starvation, a Dutch pediatrician named Willem Dick noticed a highly specific, astonishing anomaly.
Right. He was treating children suffering from a chronic, wasting diarrheal illness.
Yeah.
Doctors had called it celiac disease since the 1880s, but nobody knew the actual biological cause.
Nobody had a clue?
But during the blockade, when wheat flour became completely unavailable to the population, these dying children miraculously started getting better.
And Dick knew he had found the trigger when the war ended. Allied and Swedish planes airdropped massive amounts of bread into the country to relieve the famine, and those same children relapsed immediately.
Wow. It was a tragic but undeniably clear biological signal.
He realized it was the wheat, specifically the protein gluten. For people with celiac disease, gluten isn't just an allergen, it triggers a violent autoimmune attack.
And if we look at the mechanics of that attack, it specifically targets the duodenum and the jejunum. The immune system causes something called villus atrophy.
Right. It literally attacks and flattens those beautiful, microscopic, finger-like villi we discussed earlier.
It bulldozes the factory floor.
The entire 30-square-meter surface amplification basically collapses. The patient suffers severe malnutrition, not because they aren't eating, but because the architectural machinery required to pull the food across the barrier is gone.
It perfectly illustrates why that specific surface architecture is literally a matter of life and death.
It really is.
But even when the anatomy is perfectly intact, things can go wrong if the rhythm of the engine is off. So if this 30 square meter factory floor is perfectly designed to absorb nutrients, what happens when something else gets to the food before the intestine does?
Well, that actually brings up one of the most fiercely debated diagnoses in gastroenterology right now, SIBO, or small intestinal bacterial overgrowth.
I've heard so much about this recently.
It's an incredibly complex issue. Normally, the small intestine is a relatively harsh, sterile environment compared to the densely populated colon.
Okay.
And that sterility is largely maintained by something called the migrating motor complex. In the late 1960s, a gastroenterologist named Joseph Szerzewski helped map this mechanism out.
What does it do?
Between meals, your small intestine sweeps intense muscular contractions down the tube. It acts like a biomechanical squeegee, physically wringing out the intestine to push any residual bacteria down into the large intestine where they belong.
So if that biomechanical squeegee fails, maybe due to abdominal surgery or nerve damage from diabetes or a connective tissue disease, the bacteria migrate up and stay in the small intestine.
Yes, they multiply.
And when you eat carbohydrates, those bacteria ferment the carbs prematurely, causing massive bloating, pain, and malabsorption because they are literally stealing your food.
That is the mechanical theory, yes.
But you mentioned this is an active clinical debate. Why is it controversial if the mechanism makes so much sense?
The controversy lies entirely in how we diagnose it. The most common, non-invasive way to test for SIBO right now is a breath test.
Breath test.
Yeah, patients drink a sugar solution, and doctors measure the hydrogen and methane gases they exhale, which are produced by fermenting bacteria. But impartially speaking, the biological mechanics of this test are highly contested.
Wait, if the test just measures gas over time, couldn't a naturally fast digestion rate make it look like small intestine bacteria when it's actually just your normal colon bacteria getting the sugar earlier than expected?
That is exactly the primary criticism. Transit times vary wildly from person to person.
Oh, I see.
Standard breath tests have deeply problematic rates of false positives for that very reason, as well as false negatives. It remains a genuine, unresolved challenge in modern gastroenterology to definitively prove when someone's daily bloating is actually caused by overgrowth in the small intestine versus dysfunction in the colon.
It's a humbling reminder that as much as we've learned since the days of treating the gut like a passive coffee filter, we are still mapping the nuances of this engine.
We absolutely are.
So let's synthesize this journey for you, the listener. If there's one core reality to take away, it's that your small intestine is not a short passive pipe. Oh, and by the way, that reminds me of another massive myth we should bust right now. Fiber.
Oh, yes, the fiber myth.
People constantly think dietary fiber is absorbed in the small intestine. It's not. Human enzymes can't break it down, so it completely resists digestion here and passes straight to the colon to feed your microbiome.
Correct. The small intestine is exclusively dedicated to the active, energy-intensive extraction of sugars, proteins, and fats. It is a highly specialized 30 square meter engine.
And understanding the mechanics of that architecture dictates so much of your daily life.
It explains why oral rehydration salts can rapidly cure a hangover by forcing sodium-glucose poo transporter to pull in water. It explains why lactose intolerance causes such rapid distress. It's nothing more than a missing enzyme on that microscopic brush border.
It even dictates the multi-billion-dollar pharmaceutical industry, right?
Definitely. Drug developers have to design oral medications based on exactly which specific segment of the tube a drug needs to dissolve in to hit the right cellular transporter.
It is an absolute marvel of biological engineering. But here's where it gets really profound, and we haven't even touched on the manager of this massive factory.
The man.
We talked about those biomechanical squeegee waves, the precise mixing motions, the coordination of the valves. All of that requires immense computational power. And it's not controlled by your brain.
No, it's controlled by the enteric nervous system.
Which the neurobiologist Michael Gershon famously dubbed the second brain. Your gut is lined with its own vast, independent network of neurons. It runs the peristalsis, it manages the segmentation, and it controls this 30 square meter engine almost completely independently of your central nervous system up in your skull.
It operates on its own local intelligence, constantly reading the chemical environment and making real-time adjustments without ever asking for your conscious permission.
So we started this deep dive by saying the small intestine is the ultimate physical boundary between the outside world and the inside of your body. It's an active, highly selective wall burning energy to decide what gets in and what gets kept out.
Exactly.
But if this massive engine runs on its own complex network of neurons, firing away in the dark, entirely without your conscious brain's input, who or what is really making the decisions about what becomes a part of you? Think about that next time you sit down for a meal.
A fascinating biological mystery to leave on.
Thanks for joining us on this journey. Catch you on the next deep dive.